All right, let's talk about lipids. So lipids are going to mostly be hydrophobic, nonpolar, and contain many hydrocarbons. So looky here. Carbon, 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 and we are assuming full saturation of hydrogens between or on all of these carbons. Here you have carbon, 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 all these carbons saturated with hydrogens. Same thing here. So nothing but hydrocarbons. Hydrocarbons. You see a few oxygens here and there, but compared to the grander scheme of the rest of the molecule, a very small fraction of it. In this case of the phospholipid, you do have a lot of oxygens here, and in fact, this specific lipid is going to be amphipathic, meaning it has a polar region as well as a nonpolar region. So, like I said, your fatty acids or your lipids, sorry, your lipids are actually going to be more nuanced. It's not like they're all composed of the same subunit, though you will see some motifs when it comes to the description of, triglycerides that are composed of fatty acids, and if you compare that to phospholipids. So they're generally defined as a substance that is insoluble in water. Insoluble. Why is that? Because it's nonpolar, therefore hydrophobic. Therefore, it doesn't mix with water. So it's insoluble in water and soluble in organic solvents like ether and chloroform, for example. So other nonpolar stuff will dissolve it pretty easily. All right, so let's check out triglycerides. Triglycerides. So those are going to be your fats and your oils. Their main function is the storage of energy, and your triglycerides are going to be composed of one glycerol molecule, so that's this guy here, one glycerol molecule, and three fatty acid chains. Triglyceride, tri meaning three. So we have three fatty acid chains and one glycerol backbone. So here we have the nonpolar chains, or these chains are nonpolar. The larger circles are carbons and the smaller circles are hydrogens. So nonpolar chains of carbons with abundant hydrogen. And they are linked to glycerol by ester linkage. So this fatty acid is bound to this glycerol by an ester linkage. This fatty acid is bound to this glycerol by an ester linkage. And this fatty acid is bound to this glycerol by a ester linkage. So here, let's talk about the difference between saturated fats and unsaturated fats. Your saturated fats are going to be fully loaded with hydrogens, therefore having no double bonds. So no double bonds between each of these carbons here, and they're fully loaded, fully saturated with hydrogens. Now your unsaturated fats are going to have at least one double bond. And if you have one double bond, well, that's one less bond on each of these carbons that could be potentially bound to a hydrogen. Therefore, it's less saturated with hydrogen. Therefore, we call it an unsaturated fat. So it has at least one double bond replacing two hydrogens, and normally what that's going to do is create this little kink in the fatty acid tail. So here we're just looking at the fatty acid tails that would be composing your triglycerides. They would be part of your triglycerides. So monounsaturated fatty acids have only one double bond, whereas oftentimes what we'll see is more than one double bond on our fatty acids. So polyunsaturated fats are going to have many double bonds. So we have a polyunsaturated fat here, polyunsaturated fat here, and if you count from the tail, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, that's what makes the omega number. That's how we define the omega number. So from the tail, how many carbons in from the tail is the first double bond? Well, the sixth carbon is the start of the first double bond. Therefore, we constitute this linoleic acid an omega-6 fatty acid. Linoleic acid. Okay, so in this case, you have carbon 1, carbon 2, carbon 3, forming an omega-3 fatty acid. All right. So yeah, the omega number is the number of carbons from the tail with the first double bond. All right, so monounsaturated fatty acids. Some examples of monounsaturated fatty acids are going to be things like olive oil, canola oil, peanut oil, avocados. Some examples of your polyunsaturated fatty acids are going to be things like safflower oil, sesame oil, soybean oil, corn, sunflower oil, nuts and seeds. And then you're going to find an abundance of omega-3 fatty acids in your canola oil, soybean oil, flaxseed oil, and also your fatty fish. So the thing is, each oil is going to be a mix of different fatty acids. So mostly would apply here. In your olive oil, it's going to mostly consist of monounsaturated fats, though it will have some polyunsaturated fats. It might even have some omega-3 fats. Your avocados will mostly have monounsaturated fats. Your corn oil will have mostly polyunsaturated fats. All right, so let's talk about trans fats. So trans fatty acids. A trans fat is going to be an unsaturated fat, meaning it has a double bond somewhere, though it behaves like a saturated fat. So because of the arrangement of carbons, it makes it behave more like a saturated fat. So here we're looking at a saturated fat, fully saturated with hydrogens, and then in this case, you have a unsaturated fat with more than one. This is a polyunsaturated fat, but it has a cis double bond between these two carbons. And cis actually means same. That roughly translates to same. And so if you notice here from this carbon, or you could say from this double bond, this carbon moves down at that angle, and this carbon moves down at the same angle, just in the opposite direction. So this is a cis double bond because both of these carbons move down at the same angle, just in different directions. Now, if you look at this trans double bond, you would kind of have to like rotate this molecule going up, but from that double bond, one carbon goes in the downward direction, whereas the other one goes in the upward direction. So structurally, what happens is this molecule here will behave more like a saturated fat. However, our body or our cells identify this as a unsaturated fat. Now, typically, your cis double bound unsaturated fats are a little bit more jiggly. They're a little bit more fluid. They have a lower melting temperature, whereas our trans fats are more solid, more rigid. And so if our cells are identifying an unsaturated fat and plugging it in where an unsaturated fat should go, however it behaves like a saturated fat, that could lead to a little bit of confusion within your cell. Not really confusion, but just bad things could happen. All right, so let's talk about the degree of saturation. Well, the degree of saturation, so the more double bonds you have, that's going to determine your fluidity and melting point. So usually the more polyunsaturated it is, the more fluid it is, the lower the melting temperature. Now, typically your saturated fats are going to be solid at room temperature, and they're going to have a very high melting point. So you're going to typically find your saturated fats in animals and waxes, for example, solid at room temperature. Now your unsaturated fats, those are typically going to be liquid at room temperature, and they're often found in most plants. All right, so we're moving away from triglycerides, and we're moving on to phospholipids. So phospholipids, like triglycerides, they're going to have a glycerol backbone. They're going to have fatty acid tails, but 2 of them instead of 3. So you have 2 fatty acids. Where does the third one go? Or what's replacing the third one? What replaces the third one is this thing called a choline molecule. So if you look here, we have this polar choline molecule. We have a glycerol backbone, and instead of 3 fatty acid tails, you have 2 fatty acid tails. And so our phospholipids are considered to be amphipathic, meaning it is both polar and nonpolar on the same molecule, just one region of the molecule is polar and the other region of the molecule is nonpolar. All right. So phospholipids, I forgot to mention, phospholipids are going to be the main component of our cellular membranes. We might have heard the term phospholipid bilayer before. So here is an example of a phospholipid bilayer where the blue heads are the polar choline area, And then the little fatty acid tails are these little black lines here, or this magenta area. This is going to be your nonpolar or hydrophobic area. And then this is going to be the exposed hydrophilic area, the polar hydrophilic heads of the phospholipids. So your phospholipid head is going to be polar, therefore hydrophilic, and your tails are going to be nonpolar, therefore hydrophobic. And then these little green things, those are probably little cholesterol molecules. All right, so let's talk about our steroids. So steroids are typically going to have a ring-like structure. So here we just have, hydrocarbons, but in the form of rings. So things like cholesterol, which is what this is, bile, sex hormones. So our sex hormones are actually created using cholesterol as a precursor. So they're synthesized from cholesterol. Same with adrenal hormones. And so typically our steroid hormones or steroids are going to be found in animal products like meat, eggs, fish, poultry, and dairy. So steroids, or you could say, your steroid hormones are types of lipids in a sense. Now we have other compounds that are kind of associated with lipids. So we have carotenoids, which are light-absorbing pigments, which are fat-soluble or lipid-soluble. Same with or things like beta-carotene. And then our bodies will actually use vitamin A. We'll use beta-carotene to create vitamin A. So your vitamins A, D, E, and K are all fat-soluble. And then lastly, we have our waxes. So these are going to be things that your birds will use to coat their feathers. That actually makes it waterproof. Look at that. It's waterproof because it's hydrophobic, right? Or not waterproof because it's hydrophobic. The water kind of groups together. The water is afraid of the polar stuff too, or the nonpolar stuff, and the nonpolar stuff is afraid of the water. And then you have leaves and stems. So you'll coat some of your stems and leaves with waxes, and this is actually to prevent the evaporation of water too, to kind of make it impermeable to the water inside of the plant, which it uses for photosynthesis. And then same with honeycomb. This is another type of lipid. All of these are types of lipids that are all hydrophobic.